and my name is Csete, and unless you're Hungarian, you have no way of knowing how to pronounce that name. And I'm an associate professor in the anesthesiology department at Emory, where I'm in charge of the team that does liver transplant anesthesia. And I'm an assistant professor in cell biology at Emory, and now a faculty member in the program in neurosciences and the program in biochemistry, cell, and developmental biology. And I'm hoping also to become a faculty member in the bioengineering program. So today I'm going to tell you about the history of the work that started first at Caltech when I was the world's oldest graduate student in the late 1990s. After being a tenured professor in anesthesiology at UCLA, I went back to become a lowly graduate student at Caltech. And the work started there and is now being carried on at Emory. We have our lab space in the dental building, which is the building that has the medical library in it. There's no dental school there. So we're 4 floors above the medical building, and those are some contact information data for you. My email address on the Emory website is wrong. So I'm going to just try to get us all on the same page and tell you about oxygen in relation to cell culture and why I think gases are extremely important but ignored in biology when we model our diseases in a dish. So to get us all on the same page of oxygen concentrations, remember room air is 21% oxygen, and the highest concentration in your body is in the lungs. As room air comes in, the alveoli are 14.5% oxygen. Arterial blood is 12% oxygen. Venous blood is 5.3% oxygen, and the mean tissue in your body lives at 3% oxygen. So adult tissue basically varies between venous and lower, and if you have an arteriolar blood supply, you may live a little bit above venous tensions depending on the local blood supply. The brain, which you'll hear a little bit about today, across mammalian species, the brain lives at 1.5% oxygen at the tissue level. Many cells live happily below that, and here we are cultivating our cells in 20% oxygen. Basically an order of magnitude off from what is seen physiologically, and that's the adult. So in embryos in which stem cells are very enriched, the pre-implantation embryo is dependent for oxygen by diffusion, and even after implantation, in the first trimester, the trophoblast really doesn't erode in a way to give a good blood supply, oxygenated supply, until the second trimester. So probably the embryonic oxygen tension through the first trimester is well less than 1% at the tissue level. And so there is considerable heterogeneity as well, and these are some mean oxygen tensions in various regions of the brain. And as someone at Caltech pointed out to me a long time ago, it's the primitive regions of the brain that are lowest in their oxygen tension. Knowing this data, I decided to grow stem cells from brain at physiologic 2% oxygen tensions versus the way they'd been cultivated for a long time in 20%. And so I'm going to follow you through the story of these cells. Our cells from rat brain are derived from E12 to E15 embryo, so late gestation phase embryo brains. And like all stem cell preparations, if you want to keep the cells in an undifferentiated state, you grow them in the presence of a mitogen, in this case basic fibroblast growth factor, and at low density. And then when you want them to differentiate, you withdraw the growth factor. And we looked for the traditional brain lineages, not for more exotic things that CNS stem cells from brain may or may not do. And the first thing that happened when the cells were grown in lowered oxygen conditions was that over the course of a 4 or 5 day experiment, there were just lots more brain stem cells, 4 or 5-fold, and this is a rare, difficult to find population, so this was a nice finding. And not surprisingly then, when you look to follow the rate of proliferation of cells in culture in low physiologic versus usual laboratory conditions, these cells are labeled with BRDU, which is a thymidine analog taken up by cells when they're synthesizing DNA in anticipation of mitosis. And then you can go in and label the BRDU. And throughout this whole culture paradigm, while the cells are still stem cells, and even later when they are neuroblasts or glioblasts, still with the ability to divide, there's more proliferation going on in low oxygen versus in 20% oxygen. And when you quantitate this, it's statistically significant throughout the culture period. We did parallel work in human CNS stem cells that were derived from a company, Clonetics. And let's see. In this case, we're looking at cells that are labeled for apoptosis with a tunnel label. And as the cells were known to do, in about 2 weeks in traditional culture, about half these cells labeled in brown here are undergoing programmed cell death, whereas when you grow the cells in lowered oxygen conditions, you can maintain them for a longer period of time with virtually no apoptosis going on. And when this was quantitated back in the rat embryonic stem cells, again, the decrease in apoptosis in lowered oxygen is statistically significant. So what you have is cells that are, you have more abundant cells when you grow the brain stem cells in low oxygen because of two independent paths being affected. There's increased proliferation and decreased cell death, both. And the apoptosis phenotype in low oxygen is something that was really striking in the small amount of work that we did with retinal stem cells in mice. So retinal stem cells are relatively easy to get from embryonic preparations in mice, but within a day after birth, they're almost impossible to find and grow. And we isolated stem cells from newborn pups at a day and then grew them in lowered versus 20% oxygen conditions. And again, we could kind of turn off the apoptosis process and keep the cells growing in culture longer than people had been able to do before. And I really think that this is a striking finding because of the oxygen story. So the major physiologic event at birth that probably hurts these cells is that they've gone from a low oxygen embryonic environment to the high oxygen atmospheric environment. So again, back to our CNS stem cells, not surprising when you look at clonal assays, which is what developmental biologists want to do, to look at the multipotentiality of cells. There were more clones in low oxygen, and the clones were of a larger size. So far I've really only told you what happens when the cells are still stem cells, undifferentiated stem cells. And please, if I say anything you don't understand, just raise your hands. Okay. So we let the cells differentiate, withdrew mitogen, and let them get quite dense in low versus high oxygen. And we got everything we'd expect the cells to generate. We got astrocytes, we got myelinated glial cells, oligodendrocytes, we got neurons. There were less stem cells at the end of a 10-day experiment in low oxygen, and lots more of everything else that was differentiated because you started with more stem cells to differentiate halfway through this experiment. And the whole process, as was obvious when you looked under the dish, of differentiation was happening a whole lot faster in low oxygen. So within 1 or 2 days after differentiation, you could see clusters of oligodendrocytes in low oxygen, whereas it took a much longer time in high oxygen conditions. And at this point I was thinking, I have an interesting way to expand stem cell populations, but maybe this is just kind of a kinetic phenomenon and not all that terribly interesting. But when we started to look at the kinds of neurons, daughter neurons, that are generated in low versus high oxygen conditions, this was really very strikingly different than what we expected. So what you see in this picture is that anything that's labeled in red is labeled with anti-beta-tubulin 3, a marker of all neurons. And anything that's labeled in green is stained with something that specifies the signaling pathway used by distinct neuronal populations. So in this case, tyrosine hydroxylase, for example. And these numbers are the percentage of the total neuronal population represented by these subtypes. So again, what we saw was a skewing of the neuronal differentiation. So there were more serotonergic neurons as a fractional percentage of the population in low oxygen, and strikingly more dopaminergic neurons, which we'll come back to, but less GABAergic and glutaminergic neurons as a fractional representation of the neuronal population in low oxygen. But whole lots more of everything, remember, in low oxygen. But we were skewing the cells towards what developmental biologists would say is a ventral neuronal phenotype. And in parallel work that was going on with Sean Morrison and David Anderson's lab at the time, we also looked at peripheral nerve stem cells from embryonic rat sciatic nerve, and we saw this same skewing of development towards tyrosine hydroxylase positive adrenergic neuronal pathway. These would be sympathetic neurons in the periphery when the cells were cultured in low oxygen. And similar to what we had found in the brain stem cells, the stem cell yield was increased, stem cell proliferation was increased, and then there was this skewing of the differentiation process. So in order to tell you how quantitatively important this change was, skewing towards a dopaminergic neuron production, I go back to compare to a paper that was in Science a few years back, in which the very same stem cells that we used were transfected with a neurotrophic factor that's now being used clinically, GDNF, glial-derived neurotrophic factor. And the paper was published in Science because with transfection of this gene product, there was an 80% increase in dopaminergic neuron yield from the stem cells. Very remarkable. When we analyze our data the same way, just by cranking down the oxygen on a bad day, you get a 900% increase. This is a very dramatic effect on differentiation. And so when you see tyrosine hydroxylase positive neurons, you want to make sure that they actually do release dopamine, and using HPLC, sure enough, there's more dopamine in the low oxygen cultures, and you can evoke the stores with high potassium in the medium, and there was more dopamine stored in these neurons in low oxygen as well. So this is an experiment that I never published, but I think is really important, and it's something that I think about all the time in our other systems as well. So I was interested in where in developmental time in the dish oxygen was having this effect on producing dopaminergic differentiation. And so we grew the cells in the presence of mitogen again as stem cells, either in low or 20% oxygen. And then at the time of differentiation, when the mitogen was taken away, half the cells were switched to the other oxygen condition, and half stayed where they started. Okay. And so what we found was that if you switched over to traditional high oxygen conditions at the time of differentiation, you didn't lose much in dopaminergic cell yield. If you switched to low oxygen from 20% oxygen at the time of differentiation, you did increase the dopaminergic neuron yield, but you never got up to the levels as when the cells were exposed in their undifferentiated stem-like state. And over in our lab, we are finding that the more primitive the cell, the more plastic it is to the changes in gases that we make in the laboratory. So I think this is an important message. So what was oxygen tweaking? The rate-limiting pathway in adrenergic signaling pathways is tyrosine hydroxylase, rate-limiting enzyme. And we found that TH was regulated at the protein level. So in low oxygen conditions, tyrosine hydroxylase at the protein level was more stable and more abundant. And we also looked a little bit at some important regulators of oxygen physiology, and I won't go into detail here unless you all want to do this at the end with questions. But the message I wanted to bring home to you here is that not only are there changes by the gas conditions in terms of protein stability and you'll see protein modifications. But we found huge changes in protein trafficking. So, for example, the von Hippel-Lindau gene, when you stain in the brain stem cells, in 20% oxygen is found in red, both in the nucleus and in the cytoplasm, whereas in the low oxygen conditions, it was found exclusively in the nucleus. And obviously, the kinds of function proteins have is dependent on their partners, and they're going to have a different repertoire of partners in the nucleus than they can find in the cytoplasm. And similarly, Col2, which is a partner of the von Hippel-Lindau gene involved in responses to oxygen, was also trafficked in and out of the nucleus in a similar fashion as a consequence of the gas conditions. So at the message level, we then were asking what in the traditional dopaminergic neuron developmental pathways is being changed by changing the oxygen tension. And so we did a series of semi-quantitative PCRs here so that all you really need to know about this picture is that the size of the white blob is proportional to the amount of messenger RNA for that gene. And you could see some striking changes. So FGF8, for example, was really abundant, fibroblast growth factor 8, at the end of expansion of stem cells in low oxygen, and there's a very faint band here. And we found other changes as well. Surprisingly, tyrosine hydroxylase was not regulated at the message level, as the literature would have suggested, and the traditional maintenance factors, growth factors for dopaminergic neurons, again now being used clinically, were not changed at the message level. And when we saw these kinds of changes, if we could get our hands on a protein, we threw the protein on our 20% oxygen cells to see whether this protein recapitulated the effects of low oxygen in promoting dopaminergic differentiation. And FGF8, among others, did not. But we found that it was a pretty good mitogen for the stem cells, which we hadn't known. So those were genes that are known to be important in the embryonic development of dopamine neurons, and we didn't really find any players that were related back to our low oxygen story. But as many of you in the room probably know, the classic oxygen-regulated gene is erythropoietin. Erythropoietin is a growth factor for red cells when you're anemic. It's produced in the kidney, signaling stem cells in your bone marrow to make red cells. And we found that erythropoietin was more abundant in our low oxygen cultures than in our 20% oxygen cultures, and really almost as an afterthought. As a control experiment, we added erythropoietin to our brain stem cells and found, much to our surprise, that this actually recapitulated much of the effect of low oxygen in promoting dopaminergic neuron differentiation. And in fact, when we added blocking antibody to EPO to the cultures, we could block the production or differentiation of these neurons in both oxygen conditions. So these results that I just showed you were published basically the same month as a group at Rockefeller showing that erythropoietin may be globally neuroprotective in injuries, very gross injuries like stroke and crush, head injuries. And so there now is a field that's exploding looking at erythropoietin as a potential therapy for things like strokes and traumatic injury to the brain. We're also following this up in a separate series of experiments to look at erythropoietin as potentially a novel protective therapy for Parkinson's disease, because as I showed you, our major effect was in neurons that are dopamine producing, and these are, in fact, the neurons that are lost in patients who have Parkinson's disease. So as a plug, just to say what's going on in the lab, potential projects for students, we have a dopaminergic neuron cell line that is being used as a cleaner model than the mixed cultures we derive from stem cells to look at the biologic effects of erythropoietin on these cells. And luckily, they all express abundant erythropoietin receptor, just like dopaminergic neurons in the adult rat brain appear to in our hands. So if you want to take this from stem cells in a dish to a clinical scenario, you have to look at animal models of the disease you're interested in. And for Parkinson's disease, there are 3 models now available to us. All of them are flawed in some fundamental way, but they are useful nonetheless in screening for protection with novel drugs, in this case erythropoietin. So Mptp toxicity is the first model that we looked at very briefly. Mptp was a toxin that was found as a contaminant of heroin in San Francisco when the addicts started to become acutely Parkinsonian, and became the standard model of Parkinson's disease once it was isolated from the heroin. The other model that we've looked at is direct injection of 6-hydroxydopamine into the brain using stereotactic coordinates. And the hydroxydopamine causes a local oxidative stress that causes degeneration of the dopaminergic neurons locally. An insecticide in high doses can also cause degeneration of basal ganglia neurons. We haven't used that model yet. So let me walk you through a little bit of our results in the mouse and the rat model. So we used high-dose Mptp in mice, preceded a day before by either erythropoietin given peripherally in the peritoneum or stereotactically into the brain. And then we sacrificed the animals at a week and looked at the tyrosine hydroxylase positive neuronal population in their brains. We also had the animals scanned occasionally, and there was good dopamine uptake by these PET scans, suggesting that we hadn't changed the lesion very much by our EPO treatment. And indeed, when the graduate student who did this work started counting the number of neurons in lesioned animals versus lesioned animals that had been pretreated with erythropoietin, she didn't see a huge difference at a week. But what I didn't tell you is that she was only able to count a subfraction of the animals that we treated, because what we found, and the numbers are now larger, was that erythropoietin was very protective for survival in this model. So we had purposefully picked a high dose of Mptp. A lot of the animals died within a day and couldn't be looked at a week later. And we were seeing this striking difference in survival. So Elise Sheboygan, who did this work, is now following up for her Ph.D. work back at Michigan, looking at why it is that erythropoietin is protective. And we, at this point, probably think that this is an effect on the glomus cells in the carotid, also dopaminergic cells that drive respiration. In the rat model, which is ongoing in the lab here at Emory, I'm going to show you some pictures of what the brain looks like when it's treated with 6-hydroxydopamine on one side. So here, the brains are anesthetized, the rats are anesthetized, their heads are put in a holder, and using stereotactic coordinates, we inject the 6-hydroxydopamine into the right side. The day before, the animals either receive erythropoietin or saline. So here's what the lesion looks like, and you'll be marching backwards through the brain. The substantia nigra on the left stained for tyrosine hydroxylase producing neurons, and here at this particular slice, this lesioned animal, we really can't find much in the way of neurons. And as you march back, you look for asymmetry, and there is, in most places, a striking asymmetry with loss of the neurons on the right side. So I'll march you through. And towards the back of the brain, we pick up more neurons. Again, by PET scan, there's an asymmetry of the lesion. And this is what the animals look like who receive EPO, pretreatment, again, stereotactically at the same place that they're lesioned. Again, you're looking for asymmetry. The right side was the lesion side in the animals, and marching backwards. So we're not through the quantitation yet, but there's a striking difference. And even in the behavioral assays, very simple behavioral assays that you do to look at the integrity of the dopaminergic pathways, it's clear that erythropoietin, if you get it to the right place at the right time, is highly protective in this artificial Parkinson's model. Unfortunately for us, when we give erythropoietin peripherally, it doesn't cross the blood-brain barrier in this model, unlike in strokes where the blood-brain barrier may be disrupted and EPO can get through. So we're now working on ways to clinically deliver erythropoietin to the brain in a way that would be palatable to patients. I think that patients, for example, with progressive Parkinson's would happily exchange a spinal injection of EPO once a month if it meant that their Parkinson's didn't progress. And so we're now looking at potentially delivering EPO to the rats by intrathecal injection. For even an anesthesiologist, that's quite a challenge, but we have it figured out pretty well. We're also looking at other ways to maybe fool the blood-brain barrier to getting this drug across. But the most important translational work that we're doing is starting up our experiments in the monkey model of Parkinson's disease. So the neurologists at Emory have been very struck by these results and are lending us their Parkinsonian monkeys for our translational work in primates. And this is also work that requires more students. So now that I've told you about how we got from basic changes in oxygen in brain stem cells to something that I think is clinically very relevant, I'm going to tell you a parallel story about muscle stem cells. And again, some very basic biology that's changed by the oxygen tensions, and then where we're going with this, again, clinically. So as you sit here today, probably the most abundant solid organ system stem cell that you've got in your body is in your skeletal muscle. About 1 in every 1,000 nuclei is a stem cell that sits in a very protected environment in the muscle between the basal lamina and sarcolemma. And these cells are called satellite cells because of their appearance, which you'll see. Now, they're usually quiescent until you go out and wear and tear your muscle enough to cause them to come out and have to regenerate new muscle. And just like the marrow cells that replace blood elements throughout life, these are the cells that replace your skeletal muscle throughout your life. And they have a high proliferative potential, just like other stem cells. Until this work, people thought they could only make muscle. So the model that we use here, one of the models that we use, is to take single isolated muscle fibers from adult mice. And the act of putting the single fibers into culture causes the stem cells to emerge from that protected basal lamina environment, come out and divide on the fibers. And here's a cluster of probably 16 cells still hanging by basal lamina. And then they will migrate off the fiber. Actually, they just dramatically tear away from the fiber. It's really quite violent. And they'll lay down myoblasts, the next stage in muscle development, on the bottom of a dish or on a parent fiber or on a dead fiber. And the myoblasts will fuse into contractile, twitching, functional myotubes, muscle tubes. And this is a view of muscle in cross-section. And you can see that the stem cells are small and they're surrounded by basal lamina. That's still the best way to identify them. The molecular markers are a subject of a lot of controversy. Okay. So after what I told you about the brain stem cells, I'll walk you through this very quick because it's not a surprise. Just like the brain stem cells in low oxygen, when you grow these cells in something that's more physiologic and look for proliferation, it's increased in low oxygen. These cells do not undergo apoptosis while they're still on the fiber, so there was no difference in apoptosis between low and high oxygen. But there was a difference in differentiation. So muscle developmental biologists have worked out the core regulatory pathway of genes that promotes a stem cell to become a muscle cell. And I don't think it's important for you to know the names of the genes. Just know that the genes that are important for developing muscle in an embryo are also the same ones that these cells use to regenerate muscle in the adult. So they were the ones that we looked for, going in and plucking the cells singly and then doing nested PCR reactions at the single cell level to see what oxygen was doing to change the gene expression in these cells. And so what we found was that as a percentage of the cells, these genes were much more likely to be expressed in a higher percentage of cells in low oxygen versus high oxygen. So low oxygen conditions cause the cells to be committed to muscle differentiation and pursue that pathway. But one of the things that's a problem in the single muscle fiber cultures is that you get contaminants. You can't do a perfect surgical dissection. You can't do a perfect isolation. And one of the things I noticed was that cells in 20% oxygen were often contaminated by fat on the bottom of the field, whereas in low oxygen, I didn't see that much. So we went back to the traditional stem cells that are used to study fat differentiation and found that when we grew them at 20% oxygen and forced their differentiation pharmacologically, that you could easily get these blobby fat cells. But growing the cells in 2 to 6% oxygen completely turned this process off. And just like there's a core series of genes that regulate commitment to muscle development, there's also a core series of genes that regulate commitment to fat. And these genes, in this case PPAR gamma, were more likely to be found in the cells in 20% oxygen versus in lower oxygen conditions. Which led me to think that my friends, the satellite cells, could also be the source of fat in our cultures. And sure enough, when we go back and stain for fat using oil red O, which picks up triacylglycerides, the satellite cells are 6 times more likely to make fat in 20% oxygen than in low oxygen. And not only that, but myoblasts, which are cells that are past the stem cell stage and have already started to turn on some of the muscle genes, these cells were thought only to be able to go on and make muscle. If you let them sit in high oxygen for long enough, they will also start to accumulate fat. And again, the molecular fingerprint is such that there's more PPAR gamma being expressed at the single cell level, at the message level, in high oxygen versus low oxygen. So what's nice about the system, again, is just like we skewed development of the neurons towards a dopaminergic fate, here, low oxygen promotes muscle differentiation and turns off fat differentiation. And so you have a prospective way of programming your stem cells to do one fate over another, and that's a way for you to study how pathways are turned on and off to promote one or the other pathway. Now, at this point, most people are asking us how much of what we see in terms of oxygen is actually an oxidative stress story. And I have a postdoc in the lab who is very interested in this, and I'll walk you through some of his early work. So is high oxygen in culture high oxidative stress in culture? Well, Jason Hansen went in and used this dye to get a read on the global oxidative stress in our muscle cultures. And this is a dye that's non-fluorescent until it's oxidized, and basically you take a fluorescence measurement, and this is proportional to the amount of intracellular oxidative stress. And over many muscle cultures, we found, sure enough, there's about a 30% increase in reactive oxygen species in our high oxygen cultures. So the high oxygen is toxic, perhaps, in this regard. So again, I still take care of patients, and the obvious clinical correlate here, where do you lose muscle mass obligatorily and gain fat? Well, and you accumulate reactive oxygen species as well. It's when we age. And in fact, from age 65 to age 80, no matter who you are, a world-class weightlifter, marathoner, or a couch potato, you lose a third of your muscle mass. And I think that even though this has been shrugged off as a non-specific degenerative phenomenon, that this very potent and abundant population of stem cells is probably playing a role in the change in phenotype in muscle. And I just last week came back from a meeting that was called by the National Institute of Aging, called the ABC study. And so I want to tell you a little bit about the meeting because it brings a physiologic coloring to the work that I'll tell you about. So quietly with your tax dollars for the last decade, the NIA, National Institute of Aging, has been conducting a study called ABC, Aging and Body Composition. And in large cohorts in Oakland, Pittsburgh, and Memphis, people have been followed very carefully for changes in their body composition as they age, including with repeated total body MRIs. And the radiologists had started just now to develop the MRI results, and that's why I got a call. They found that as a consequence of aging, there was intramuscular fat accumulation. And an epidemiologist from NIA called me and said, you know, as soon as they told us that, we kind of remembered that we fund somebody who thinks that muscle can turn to fat as you age. And it was really very striking because when you look at the MRIs, the quadriceps had much less accumulated fat than the hamstrings, for example. And the average couch potato is going to use the quads a lot more than they do the hamstrings. And in a parallel study that's going on in Finland to look at ABC with aging, there was much less fat accumulation in general because the Finnish are much less sedentary than Americans. So something about our environment and our exercise and all of this, but I think related back a lot to oxidative stress, is causing the accumulation of fat inside of muscles. And I think it's logical to assume that the source of that fat is the resident population of stem cells. So we're going on to look at how the phenotype of stem cells changes with aging as a consequence of oxidative stress. And this is a very large program project, partly at Emory, partly at Michigan, partly in San Antonio, and partly in Liverpool, England. But as you can imagine from what I've told you about stem cells, there's lots of ways that you could change these stem cells biologically so that they wouldn't efficiently regenerate with aging. They could stop proliferating. They could die. They could lose their ability to migrate to the places where muscle needs to be replaced. Again, you can change the gene expressions that reinforce muscle differentiation, or you could turn on genes that force them to be not muscle. And so this is the kind of things that we're looking at in very, very old stem cells from muscle versus young stem cells. And we're also pushing the process a little bit by looking at animals who have antioxidant genes knocked out. And again, the details of this I think are unimportant, but what we've done is generated a whole bunch of knockout animals who can't scavenge or properly process reactive oxygen species. So we were trying to make their muscle even older than it would be with their chronological age. And so what we're finding is that there's defects everywhere in the stem cells as a consequence of the environment in which they live in these aged animals. So there's a profound proliferative defect in animals that have just one of the superoxide dismutase alleles knocked out. And you can rescue a little bit of it by growing the cells in low oxygen, but not all. So there certainly is a proliferative defect. But before I told you that the wild-type cells that we grow on the fibers don't apoptose within the first week or so in culture. That turns out to be that in the wild-type aged animal, there's very little apoptosis still going on. But again, when we take away their ability to scavenge reactive oxygen species, there's a huge amount of apoptosis going on. Something like 10% of the cells, at minimum, in the cultures will die over the course of 96 hours. So we're now following up on what the mechanism of this apoptotic phenotype might be. And to make a very long story short, one of the major protectors that you have in your cells to protect you against this kind of programmed cell death is called BCL2. And in our muscle preparations, we find that there's very little to protect stem cells, very little BCL2 around. As they differentiate, they seem to accumulate more BCL2. So the stem cell is particularly vulnerable to apoptotic signals from the environment. And we're also finding that BCL2 is modified in a way that decreases its normal function, and we're also finding that it's pushed into the nucleus as a function both of our high oxygen conditions and of aging. So we're not the only ones that are finding this. I just came back from a meeting a couple months ago that suggested that BCL2 in aged neurons is also being pushed to the nucleus where it can't do its normal job. So I've told you there's a proliferative defect, there's an apoptotic defect. We're trying to look at the mechanism of the apoptotic defect. What about the differentiation in old animals? Well, again, if you use low oxygen as a surrogate for youth and high oxygen as a surrogate for age, muscle differentiation is slowed down and much less robust in 20% oxygen. When we scavenge the reactive oxygen species pharmacologically, the cells start to look a lot like low oxygen cultures. And when we add reactive oxygen species in relatively physiologic amounts of peroxides, we stop differentiation in its tracks completely. So the cells don't fuse, they don't become contractile, they don't become tubes. So Jason Hansen, again a postdoc in the lab, was interested in what the oxygen environment does to the redox status of the cells as they differentiate. And he went back to the old Nernst equation, and since glutathione is the major redox couple quantitatively in the cells, we measured glutathione and reduced glutathione by HPLC and looked at the redox status during differentiation under these different oxygen conditions. And what he found is that in the conditions that I think are most physiologic, low oxygen, there's very little shift in redox potential during differentiation. When we scavenge reactive oxygen species, the cells become acutely a little bit more reduced, and they never become more oxidized than our 6% cultures. In 20% oxygen, there's progressive oxidized redox status, and with added peroxides, this becomes even more profound. And when you look at the degree of muscle differentiation, it really follows this pattern. So if you stay in a reduced state, muscle differentiation is favored. If you have a redox state that's oxidized, muscle differentiation is blunted. And at the same time, glutathione, which you need to, again, react to reactive oxygen species, is decreased in our highly oxidized conditions. So we have more glutathione in the nucleus in low oxygen and less in 20% oxygen. So again, if you look at just gross amounts of reactive oxygen species stress, you're going to get more muscle in the lower reactive oxygen species conditions in the reduced redox conditions. And this is, again, is important because the kinds of millivolt changes that I showed you that Jason followed the muscles through differentiation are exactly the kinds of changes that you need to alter transcriptional processes. So basically every week in Cell or Science or Nature, there's an article that says redox regulation of X transcription factor. And I think that people are kind of missing the point. When the redox status of the cell changes, the entire expression fingerprint of the cell changes. And this is something that's becoming very apparent to us as we look at our arrayer data. So I think I convinced you that the redox environment, the reactive oxygen species environment is important for promoting muscle differentiation. Well, what about that story about fat differentiation? So again, we used cells that are completely adipogenic in their choice of daughters. Under 20% oxygen conditions, the 3T3L1s, 100% of them will differentiate into fat. But when we lower the reactive oxygen species stress, in those cells we only get 50% differentiation. the opposite effect of muscle. So what's so appealing, I'm going to skip over this, to this model is that, again, if you change the redox status of this cell, you independently affect two pathways. You promote muscle differentiation and you turn off fat. And now we have a lot of microarray data, among others, where we've identified 21 potential regulators that would negatively impact fat differentiation, and we hope help muscle differentiation in an effort to try to identify pharmacologic tools that can help muscle regeneration. And this is now, again, a project in need of a graduate student. So what we're finding out by translating our 20% oxygen conditions into aged animal stem cells is that, first, the stem cells, again, are really exquisite sensors of their environment. And gases and reactive oxygen species are somehow sensed, but they certainly generate specific signals that change the whole profile of the cell. When we look at our arrayer data, the cells grown in low oxygen conditions just don't look like the same cells grown in 20%. There's a huge number of transcriptional changes. Protein stability is changed. Proteins are modified in these conditions. And the subcellular localization of proteins, as I showed you, is altered as well. What we think we're seeing in our early experiments from the very old mice, and now that we have really old mice, we hope to see more, is that the threshold of stem cells for apoptosis in aged animals is reduced. Their stem cells are more vulnerable to stress, and that there is an increased tendency of these cells to undergo fat differentiation, and that's a pathologic response. So let me tell you about the lab as it stands right now. Maren Fuentes is a Ph.D. student who moved down here with me from Michigan, and she's actually working on something very close to my clinical life. It's clear to me that what we're learning about why muscle stem cells decide to become fat is also going to be really important about why liver decides to become fat under a variety of clinical circumstances. And so Maren is looking at a model of hepatitis C virus-induced fat production in the liver and trying to characterize the molecular mechanism of this transdifferentiation. Jason Hansen's a Ph.D. in the lab who is interested in the oxidative stress angle to the muscle-fat fate choice. Marcus Klaas is an anesthesiology postdoc in the lab who's actually working on a chronic pain model, and he's joining the faculty in July. Missy Wilcox is a technician in my laboratory who's working on the Parkinson's project. And this is our source of support. So I'd be very happy to answer your questions.
That was a really lovely presentation. I have a comment to make that in aging, the bone marrow fills up with fat as well.
Absolutely.
I'm wondering if it's the same reaction, but I'm a chondrocyte person, and low oxygen tension is a classic way to get muscle to go into chondrocytes. Did you look for cartilage in any resection?
Yeah, we have. It takes a little longer to get bone out of these particular stem cell populations, and I'll add to what you just said. So in
Cartilage specifically.
we haven't looked for cartilage. We have found some cells that we think are early bone, and one of the graduate students in the lab, who's a rotating Emory student, I don't know if she's going to stay or not, is looking at the expression of early bone markers at the single cell level as a consequence of oxygen conditions. So we'll have some data about that real soon. Haven't looked for the chondrocyte story as well.
That's the one you're going to find, because that's the one that's affected by low oxygen.
Yeah, I should talk to you later about some ways to look at them when it's not morphologically obvious. And you're absolutely correct. I mean, the other thing that, again, from my clinical life is striking, when you look at conditions in which the satellite cells have to come out and proliferate and try to replace muscle. So muscular dystrophies is the classic. They come out, they try like crazy to make new muscle. They can't because they're missing a structural protein. Well, very often in the center of these proliferative foci, you'll see bony spicules. You very often see fat. So the environmental stress, the abnormal environment, the pathologic environment causes these fates to be taken in vivo as well. Yeah. Anybody else? Yeah.
I'm interested in the link between EPO and oxygen. So does EPO recapitulate every aspect, the proliferation, the slowing of apoptosis? Can you explain the responses based on expression levels of EPO receptors?
Okay, yeah, so we've done a fair amount of work in the N27 cells, this dopaminergic cell line. And the effect on proliferation is not profound, but these are transformed cells, so I really don't know what to make of that. It is potently protective against apoptosis in these cells induced by 6-hydroxydopamine, so we're in parallel to our animal model, we were adding 6-hydroxydopamine to the cells, and also staurosporine. So it's definitely anti-apoptotic. Mechanism I can't tell you, except that the EPO receptor is not upregulated in low oxygen conditions or when we add EPO to the cells. It's either sustained or maybe even a little bit decreased at the protein level by Westerns when we add EPO to the cultures.
What if you hit the muscle cells or the fat cells with EPO?
That's a very good, in last December, Constance Tom Noguchi's lab at the NIH published a paper, and I can't believe I missed this considering the context, showing that EPO receptor is expressed on the satellite stem cell. It's a good marker of the stem cell state. It's lost when the cells become myoblasts. So it's really quite specific to the stem cell state. We haven't used it to change the biology of the stem cells, but we're using it to mark the stem cells, that picture that I showed you in cross-section, so that we can pick them out using laser capture to try to make a satellite library, because we really have to know a little bit more about what the cells express. And I think, to my eye anyway, this is the most specific marker, the EPO receptor, for the satellite state. So it's going to be important in other stem cells. That's right. Anyone else? Thanks a lot.